The question comes up, how similar are we genetically to, say, the chimpanzee, which is apparently the ape most similar to humans that's living. And we first of all have to realize that only a small portion of the genome is devoted to coding for proteins. And it's that portion that is relatively similar. Maybe 4% of the total genome is coding for proteins. We called, at one time, evolutionists called the rest of the genome junk DNA. That was code word for we're not sure what it does, so it must not do anything, or we would know. It's more of an ego problem than it is a knowledge problem. It's been argued we're, 99% similar to the chimp in terms of that 4% of the genome coding for proteins. Others say more like 95%. Even if there was only a 5% difference, that would still be millions of base pairs difference between the two. But it turns out there are different ways of calculating similarity. And what evolutionists have done is to take that portion, that 4% that codes for proteins, and to look for similar sequences. And in the similar sequences, the question is, how different are the similar sequences, ignoring the dissimilar ones? So you can come up with figures, relatively high percentage, if you just look at those parts of the genome that are in fact similar, ignoring those that are quite different. The other thing to bear in mind is when you're coding for proteins, we have about 100,000 proteins, some would say up to 500,000 different proteins in our body. And I suspect the chimp probably has a similar number. Chimp has hemoglobin, we have hemoglobin. Krebs cycle enzymes would be similar in the chimp and in human. So it doesn't surprise me that if you're just talking about coding for proteins, there would be a similarity. Hemoglobin is similar among creatures that have red blood. Muscle protein, myosin, actin, all relatively similar. But all the different proteins don't make an ear or make an eye. It's the three-dimensional architecture of our body that goes well beyond just simply coding for a lot of protein like hemoglobin. And here, I think we get into that other 96% of the DNA that we're finding as having a lot to do with controlling how DNA is read. The whole question of the genome of living things in general is becoming very complicated. They now believe it's essentially a four-dimensional code. The basic sequence of bases in the DNA, and then the control of which portions of the genes are read. We know that they're now read in packets or pieces, not as one whole piece. And then the three-dimensional relationship of the parts of the DNA molecule in space approximates some things and keeps other things distant. And finally, time. How a gene works depends on when in time in the life history of the creature that the gene is expressed. So to think you could go into a system like this, it's four-dimensional, and make some random changes and get improvements, I would think perhaps more likely that you could kind of push things around inside of your computer and hope to get some improvements.